making in response to the low availability of these micronutrients. And what we can see is that this results in smaller percentage of their proteome being devoted to important things for growth, like photosynthesis. So at the beginning of the season, we can see that about 40% of the proteins that diatoms are producing is devoted to photosynthesis. And towards the end, a much smaller proportion of that diatom proteome is devoted to that important process of photosynthesis. So what we're seeing here is some trade-offs in protein expression, whereby the diatoms that are growing here, they need to change the way that they're growing in order to cope with this micronutrient stress towards the end of our season here. So what I've shown you here is that we can use proteins in order to understand which micronutrients are important for controlling the growth of phytoplankton in the Southern Ocean. So by demonstrating that both iron and vitamin B12 at the same time are impacting the growth of phytoplankton, we can say that these interactions between phytoplankton and bacteria, because the bacteria are the source of this key micronutrient, vitamin B12, these are critical for understanding how much phytoplankton growth is occurring in the Southern Ocean now, and what we can expect for phytoplankton growth moving into the future. So we have a lot of questions remaining about these interactions and how they're going to respond to change. So we don't know if future change in the ocean is going to induce changes in the way that phytoplankton produce organic matter or the quantity of organic matter that they're producing. And because we don't know that, we don't know how this is going to impact the growth of these heterotrophic bacteria here. And we don't then know how much vitamin B12 we expect those bacteria to be producing. So we have a lot of work to do to uncover the mechanisms that are controlling these different interactions here. But what we do know is that these two micronutrients are key to those interactions. So lots to do to understand and predict the responses to change. And here, I just wanted to show you a little bit of information about what we're predicting for change in the Southern Ocean, to emphasize why it's important that we get a more mechanistic understanding of these interactions in order to be able to predict the responses to change. So what I'm showing you here is the change in temperature that we're predicting based on model experiments in the Southern Ocean from the 1950s all the way to 2250 here. So temperature is shown in black, and ice cover, predicted change in ice cover, is shown in this blue and red here. And what we can see is that by decreasing the expected decrease in ice cover, we suspect that that's going to increase light availability to phytoplankton. And this increase in temperature, we think that will also benefit phytoplankton growth. So what researchers are predicting is going to happen in the Southern Ocean is that we're going to see a pretty profound increase in the amount of phytoplankton growth. These are just two metrics of phytoplankton growth that I'm showing you here. But what these model predictions do not take into account are these microbial interactions that I've shown to you are important for controlling how much phytoplankton grow. And so what that means is that there are important uncertainties about the interactions that we know are critical for controlling phytoplankton growth and how those interactions are going to respond to these big changes that are coming for the Southern Ocean ecosystem. One thing that I think is kind of exciting about the work or the kind of data that we've gathered in time series like these is shown here. So this is just another view of the same kind of data that I showed you before, but instead of telling you the names of these different proteins, I'm just showing you whether or not we have a predicted function for those proteins or not, whether we have any idea what that protein might be doing in the environment or not. So proteins that we do have a predicted function for are shown in gray here. And those proteins that we don't have a predicted function for, we have absolutely no clue what this is doing in the environment, are shown in blue here. And again, protein abundance is shown on this x-axis. so from low to high. And the fold change but from week 1 to week 3 is shown here. So those proteins that are out here that are blue, these proteins are proteins that we have no idea what their function is. And we know, but we do know from these data that they're very abundant in the ocean and they can change, they're dynamic. So this suggests they may play some important role in how these phytoplankton are either responding, how these phytoplankton are responding to this shift in micronutrient stress. And one thing that I really like to think about here is that I'm wondering, does the key to predicting ocean responses to change lie in identifying the function of all these unknown proteins here? So this is a really exciting intersection between biochemistry and ocean science that I think is a really fruitful area for future research. So what are the proteins that are changing across this timeline here that are really key for driving those interactions? And how can we unlock those to understand those interactions better? So to sum up what we've learned, we've seen that interactions between phytoplankton and bacteria that are driven by micronutrients can help control Southern Ocean phytoplankton growth. We've also seen that phytoplankton change the allocation of their protein resources in response to nutritional status, that we can measure those changes in the ocean, and this can tell us about the phytoplankton relationship with micronutrients, which is of value. But we're left with some really key remaining questions, and two of those in my mind are, the first one is how will the phytoplankton recipe change? Is there a chance that the amount of these nutrients that phytoplankton need is going to change as we move into a warming world? And how will these important microbial relationships change? So I want to give you a little preview about some work that my group is doing now about understanding how that phytoplankton recipe is going to change as we move into the future ocean. And that's summarized here in this figure. So what we've done is grown some diatoms in the laboratory, and we've changed the temperature at which we grow them, and we've changed the amount of iron that we give them. And so we can figure out the relationship between iron availability and growth in these plankton. And we can plot the maximum growth rate as a function of temperature. And this one's not surprising. We can see that as temperature gets warmer, these phytoplankton are growing faster. But what was a surprise to us, and I think has some important implications, is that the amount of iron that these phytoplankton need to grow at half of their maximum growth rate is much higher at low temperatures than it is at high temperatures. So what this suggests to us is that Southern Ocean diatoms might need less iron as the ocean warms. And the reason why this is important, we can think about that by going back to these predicted changes that we're expecting in the Southern Ocean. So we know that the temperature is predicted to go up, so we're expecting that alone to increase the amount of phytoplankton growth. But if we also have this need for less iron, and we're still getting the same amount of iron delivered to the ocean, we might expect that this increase in phytoplankton growth could go up even more. So I'm suggesting that we need to do more work to understand this possible relationship between increasing temperature and decreasing the need for iron in a phytoplankton cell. And what remains totally unclear is the role of interactions in these processes. So in order to start getting at the answers to these questions, these remaining questions, I was really lucky to be able to go on another research cruise down to the Southern Ocean, this time to a different part of the Southern Ocean, to the Weddell Sea, where I was able to perform some more of those incubation experiments, but this time we had the chance to change temperature as well. And so I went down with a bunch of collaborators from Germany and from the Netherlands, down to this region of the Southern Ocean and the Weddell Sea, where we conducted a series of experiments where we changed the amount of iron that was available to phytoplankton cells. And as I said, we changed the temperature that they were exposed to. These experiments are always laborious, but they're really fun to conduct. So we're wear these crazy suits so that we don't contaminate our samples for the trace metals that we're trying to do analyses on. And in this case, we used large volumes of water to do our experiment. We needed to carry them around in these crazy contraptions here so that we can collect enough phytoplankton biomass to do a lot of protein measurements on, to get a sense for how these communities are changing. And again, we incubated these experiments on deckboard incubators. This time, these are fancier and we had to control temperature, but we're still exposing them to their natural light. So this, we're still analyzing the experiments that happened at sea here. But what I'm really excited to see is if we can identify how those important interactions between phytoplankton and bacteria are changing in the face of changing temperatures there. So it was a real privilege to get to work with a big team of researchers to do these experiments and to see these gorgeous places. And I think really importantly to appreciate the algae for all that they do for us. So this image shows some sea ice algae and some phytoplankton together in the Southern Ocean. You can just see all that green and how impactful they are. So that's why I like to show this image. But I'd like to conclude by thanking my fantastic lab group. The data that I've shown you today was generated by those members whose names are bolded there. I've also got a really fantastic team of Southern Ocean collaborators. I didn't have time to tell you about my Arctic and North Atlantic work, for which I have a whole different sets of fantastic collaborators. And I also need to thank these funding sources. But I would be very pleased to take any questions from you. We'll leave you with our impactful algae here.
Great, thank you so much, Erin. That was a lovely talk.